three-dimensional shape of alpha helixes and beta-fluted sheets that are folded on each other to make a globular protein. There'll be a certain area on that protein that myosin can attach to. So a shape that myosin can fit on, and also a charge attraction that myosin will bind to. The actin is put together into a filament, sort of like stringing beads on a piece of silk or wire, and then that forms a filament, and then the filaments, two filaments are twisted on each other, and that's what you get here as the actin alpha helix. So the binding sites are twisted around this molecule, and then there are going to be 6 actins around each myosin. Myosin has heads coming out in all directions. So with these 6 actin filaments, the myosin can interact from all different angles on these actin filaments. The binding sites of actin will be covered by tropomyosin. The tropomyosin will cover about 6 to 7 actin binding sites, and then another tropomyosin will take over for the next 6 and 7 and so forth. Each tropomyosin will have a troponin molecule attached to it. Troponin has the binding site for calcium. When calcium binds, the positive charges on calcium change the shape of troponin, which then tugs on tropomyosin and moves tropomyosin off the binding site and into the little valleys between the helical filaments of actin. So here's your blocked actin binding site. Here's the binding site on myosin that wants to attach to actin. There is already bound in this relaxed state ADP and PI. It was ATP, but the ATPase in the head of myosin changes it, cuts the bond, but keeps the PI, the inorganic phosphate, and the ADP bound to myosin, waiting until the actin site is open. It's going to need calcium from the sarcoplasmic reticulum to bind to troponin to move tropomyosin off the actin binding site. So if there's no action potential at the neuromuscular junction, then there is no release of calcium from the sarcoplasmic reticulum. There will be no change in the position of tropomyosin and no interaction of myosin and actin. In the presence of calcium, calcium binding to the C subunit, troponin C, then the tropomyosin will move off into the little valleys between the filaments of actin. And as soon as the binding site on actin is exposed, myosin will latch on. And the hinge region will bend. The actin is then pulled one actin length past the myosin, and myosin will stay attached until a new ATP binds. As soon as the neck flexes, PI is kicked off, and then ADP is kicked off, and nothing will change until a new ATP binds. When the new ATP binds, the myosin will detach and go back to its relaxed state.
Troponin consists of 3 polypeptide units. One can bind to tropomyosin, one to actin, and a third to calcium. When calcium binds to troponin, the troponin-tropomyosin complex moves aside to expose cross-bridge binding sites, and cross-bridge binding occurs. The binding of actin to the myosin cross-bridge triggers a power stroke that pulls the thin filament inward.
Okay, so one, we come back.
Power stroke that pulls the thin filament inward.
One globular subunit length past the myosin. So actin, the flexion of the neck region, one globular subunit distance is pulling the Z disc towards the middle. Myosin stays attached till a new ATP binds. When ATP binds, the myosin goes back to its relaxed state. The ATPase on myosin's head will then convert ATP to ADP and PI. Myosin will then be back in a high energy state. If there are still actin binding sites available, then another cross bridge will be formed. So this cycling will continue as long as calcium is present and as long as ATP is present. You have to have calcium to keep the binding site exposed. You have to have ATP to release myosin for the next cross-bridge formation. You run out of ATP, myosin is stuck on actin, and you have rigor mortis. When you run out of ATP, after death. And ATP not being there, your calcium will start leaking out of the SR just because there's no ATPase keeping it in there. Calcium comes out, binds to troponin, cross bridges are formed, but no ATP to release myosin, so they're stuck there. That's rigor mortis.
So actin, like on section 3, number 3, after the myosin has cross-bridge and it releases, so did you say that the actin is responsible for the split of ATP?
No, there's an ATPase on. You're talking about the detachment of myosin?
No, I know that ATP comes in and lets it detach, but then I thought you said that the actin was switching.
ATPase, myosin, ATPase on myosin's head will split ATP to ADP and PI. And the speed of that myosin ATPase will determine whether you have a fast reacting fiber or a slow reacting fiber. Okay, so how do you connect the events at the neuromuscular junction to the events where cross bridges are being formed? Out at the neuromuscular junction, you create a local potential, which then creates an action potential, and when the action potential travels in the sarcolemma, which is your plasma membrane, everywhere that sarcolemma invaginates and forms a T-tubule, the action potential goes to the center of the cell. And it reaches, in the T-tubule, when it reaches these molecules that are called dihydropyridine receptors, it will activate, the action potential activates those protein receptors. They're not dihydropyridine. That's what I thought when I first saw this when I was an undergrad. They are named dihydropyridine receptors because that molecule binds to these proteins. So it's an irrelevant name, but it's relevant if you're a chemist. But physiologically, it has nothing to do with dihydropyridine. It's just that's what can bind to these proteins. The dihydropyridine receptor will be activated by the action potential. It will then link via proteins to more receptors that again have no relationship really to their name except ryanodine binds to them. So they're called ryanodine receptors. But these are channels in the SR. When DHP is depolarized, it will depolarize these ryanodine receptors and channels will open. This calcium inside the SR is at a concentration difference to the outside of 40,000 to 1. As soon as those channels open, calcium will pour out into the cytosol. Then bind to troponin, and that initiates the cross-bridge formation. So DHP activates the ryanodine channels, the channels open, calcium comes out, and you have cross-bridge formation as long as you have ATP. So neuromuscular junction, action potential arrives. You get that depolarization releasing acetylcholine into the synaptic cleft. Acetylcholine binds to the ligand receptors in the muscle membrane. That creates a local potential that if there's enough positive charge created by the local potential, then action potentials will be generated in the plasma membrane. When the action potential arrives at a T-tubule, then it can start to depolarize dihydropyridine. Dihydropyridine activates ryanodine. Ryanodine channels open, calcium comes out. So calcium released binds to troponin C, moves tropomyosin off the binding site, and myosin's head attaches, flexes. When it flexes, then PI comes off, ADP comes off, new ATP binds, the myosin detaches, there's a little space there, you've got to look really closely, there's a little space, meaning myosin detached, neck relaxes, the myosin ATPase changes ATP to ADP and PI, and if calcium is still present, another cross bridge can be formed. Cross bridges can continue to be formed as long as what? What do you have to have?
In the cytoplasm.
Calcium in the cytoplasm and ATP.
Electrical activity in a motor neuron is linked to electrical activity in a skeletal muscle cell by the events at a neuromuscular junction. Here, the axon terminal of a motor neuron is enlarged into a knob-like structure called the terminal button that fits into a depression in the muscle cell. This specialized portion of the muscle cell membrane is known as the motor end plate. Suppose an action potential arrives at the terminal button. This triggers the release of the neurotransmitter acetylcholine by exocytosis. Acetylcholine binds to its receptors on the motor end plate. The binding of acetylcholine triggers an action potential that is propagated over the surface of the muscle cell membrane. The surface electrical activity is carried into the central parts of the muscle fiber by the T-tubules. This triggers release of stored calcium from the lateral sacs of the sarcoplasmic reticulum. Now let's zoom in to see the effect of calcium at the level of a single cross bridge. The myosin cross bridges of the thick filaments contain energy stored from the prior splitting of ATP by the myosin ATPase. When the muscle is not excited, actin and myosin are prevented from binding by the position of the troponin-tropomyosin complex. When the muscle fiber is excited, the release of calcium pulls the complex out of its blocking position. Now the energized cross bridge can bind with the actin molecule. Contact between actin and myosin triggers the power stroke. The chemical energy stored within the myosin cross bridge is translated into the mechanical energy of bending. At the same time, ADP and inorganic phosphate are released from myosin. Actin and myosin remain linked together at the cross bridge until a fresh ATP attaches at the end of the power stroke. Attachment of the ATP molecule permits detachment of the cross bridge, which returns to its unbent conformation. The newly attached ATP is split by myosin ATPase, energizing the myosin cross bridge once again. If calcium is still present, the cross bridge goes through another cycle of binding and bending, pulling the thin filament in even further. Relaxation occurs when acetylcholinesterase removes acetylcholine from the neuromuscular junction and the muscle fiber action potential ceases. Calcium is actively transported back into the lateral sacs of the sarcoplasmic reticulum. The removal of calcium allows the troponin-tropomyosin complex to slip back into its blocking position. Actin and myosin are no longer able to bind at cross bridges, and the thin filaments return to their resting position.
So the only things that are missing here are local potential. So you said when acetylcholine binds, an active potential is generated, but you have to generate a local potential first. So remember that, otherwise you will miss one of the clues, one of the correct answers in the 5. The other thing is on relaxation, you have action potentials ceasing, again because local potentials are disappearing. And when the action potential ceases, then the events at the T-tubule, dihydropyridine is no longer depolarized, which means ryanodine is no longer depolarized, which means those calcium channels close. The ATPase in the sarcoplasmic reticulum is pumping all the time, but it can only pump so many calciums in, and when there's a gradient 40,000 to 1 for it to come out, as long as those channels are open, the ATPase loses. As soon as those channels close, then the ATPase starts to win, and calcium starts to go back into the SR. Then tropomyosin starts to cover the binding sites. So looking at the events, you have action potentials arriving in the transverse tubule, calcium coming out and binding to troponin. The actin and myosin start to interact. Z discs come closer together. When the action potentials cease to arrive, then calcium ATPase starts to win. The channels have closed, so calcium ATPase is pumping calcium back into the SR. Now myosin and actin do not interact. And what's the internal elastic component that helps to bring your sarcomeres back to resting level? Tighten, yes. So which band is going to shorten? I band. What about this H band? H band too. Talk about I band shortening, but since actin is going into the H zone, that is also going to decrease in size. What's going to increase in size? Not A band. The Z, not the Z disc, the other Z, zone of overlap, yes. Okay, so high energy state, myosin has ADP and PI bound to its head. No calcium present, no cross-bridge formed. Calcium present, myosin binds, PI leaves, ADP leaves, myosin stuck there until a fresh ATP binds, then the myosin releases, and you get the myosin ATPase changing ATP to ADP and PI, and cross bridges can continue to form. Yes?
When Josh had that leg cramp when he was standing up there, is that because he was like so weak and he didn't have any more ATP?
Not really, because if he had no more ATP, he'd be worse right now. We'd be digging a hole for him. Okay, so actually, probably what he did is he overworked the muscles. When you overstretch muscles and then ask them to contract, and I asked him to show off, so of course he contracted really hard, then they just spasmed. So yeah, they were fatigued a little bit, but it wasn't because there was no ATP. They're just a little bit irritable because you overworked them too hard. A lot of times you walking around, running around all day, and then you pick off your shoes and you go and relax, and you just accidentally contract your foot muscles and it grabs. You got, oh my God, or you get a cramp in the back of your leg. It's because you happen to relax, but then contract it, Manini kind contraction, and the fatigue muscle that's been overstretched, then spasm. Okay, if you run out of ATP after death, that's when you run out of ATP, then the myosin, the first cross bridges that are formed, can't release because there's no ATP available for that release, and then you get rigor, which is the sign of, usually rigor sets in a few hours after death, after ATP has been consumed, and then the calcium channels start to open, and then you have the cross bridge formation. The rigor sticks around until the proteins of actin and the proteins of myosin decompose. So the forensic scientists know that rigor is there. Death was a couple hours prior. If you take the liver temperature and it's super cold and there's no rigor anymore, then you know that the proteins are starting to denature, and that's why the cross bridge separated, came apart. It's not because suddenly there's ATP, it's just the proteins no longer can interact with each other because they're denaturing. So relaxation, you got to reuptake calcium. That is done by an ATPase, so you must have ATP present for that. Acetylcholinesterase is breaking down acetylcholine back at the neuromuscular junction. Back here, acetylcholine is breaking down acetylcholine that's on the postsynaptic membrane bound to the receptor. It's breaking it down. If it's floating free in the synaptic cleft, there's acetylcholine there as well. And in the interstitial fluid, also in the blood. Acetylcholine is not going anywhere. It's going to act in the synaptic cleft, or it's going to act nowhere. The de-recruitment of muscle fibers, when you're lowering a weight down, that requires de-recruitment. So what your brain is doing is selectively turning off alpha motor neurons. And that's allowing motor units to start to go into the relaxation phase. With de-recruitment, no action potential is arriving, then calcium ATPase wins. Calcium goes back into the SR, and tropomyosin covers the binding sites. The regulatory proteins, what are the regulatory proteins in muscle contraction? Not myosin and actin. Troponin and tropomyosin. Those regulate myosin and actin interaction. So the regulatory proteins are the ones that block the interaction. The non-contractile elastic tissue. So inside the sarcomere we have chitin. Outside, we have what are called series elastic components. These end up as being the tendons that are attaching muscles to bone. So endomysium, perimysium, epimysium. When the muscle cell ends, those connective tissues come together and form the tendon. When the muscle contracts inwards, the tendons get stretched. When the muscle is going back into, the cell is going back and the sarcomeres are going back to the resting state, this tendon that was stretched by the contraction is then going to want to go back to its resting length. It's not stretching very much. If it's stretched a lot, we'd never get our joints moving at all. But as we're contracting the muscle, there is a slight stretch to the connective tissue. And when the tension is taken away, it wants to go back to its resting length. The load that you're moving, whether it's your weight of your arm or some external load that you're lifting, this is opposing the force that the muscle has to generate to have active change in the position of the joint angle. So you can load a muscle, and as long as you don't, if you don't have the force developed to move the object, you will stay in what's called an isometric muscle contraction. If you can generate enough force to move that load, then you'll move into an isotonic contraction, where the external length of the muscle is now visibly changing, and usually we measure that at the joint angle by the change in the joint angle. So isometric muscle contraction, you do have filaments sliding past each other, but until they can exceed the load that is applied to those muscle fibers, you get no visible change in muscle length. So action potential going through the muscle. When you look at the action potential, look at the repolarization and what's happening with repolarization. If it goes right back to resting membrane potential, you are looking at a muscle action potential. What would happen at a neuron in that repolarization phase? You have an overshoot. So if you look at a neuron action potential, it has that overshoot. Muscle goes right back to resting level, no hyperpolarization. So remember that, because I might show you a nerve, I might show you a muscle, I might ask you to differentiate between the two. So you look at stimulation of the muscle fiber. Action potentials are generated, they're propagated along the length of the muscle and also into the transverse tubules. The action potential is over before you see any change in the muscle tension. Why? The action potential in the muscle is over before you see any change in the muscle tension. Why?
The calcium has to come out.
And?
You have to have enough calcium to get the total.
Has to bind to troponin, move tropomyosin.
It's a long process.
It's a process, takes time. So the action potential is over, calcium starts to come out, cross bridges start to be formed, cross bridges will continue to be formed, and you will reach the peak of the muscle tension. And by the time you're reaching the peak, what is generally happening? You change your muscle length. Why do you have a decrease in tension?
Calcium is moving back
When calcium's moving back,
into the sarcoplasmic
why?
because the action potential is high. So there's no, the local potential,
They start disappearing.
local potential goes down, and then so the
Okay, so local potential at the neuromuscular junction disappears.
So the ACTA is pushing calcium back because the calcium channels are closed.
Okay. Why are the calcium channels closed? The ACTase is winning because the calcium channels are closed. Why are the calcium channels closed? Because there's no depolarization of what?
Of those two things.
One that starts with a D, pyridine, dihydropyridine, and the R one, ryanidine. Okay, gotta remember those protein names, even though they're sort of irrelevant. That's their names. Dihydropyridine is not being depolarized, so it is not depolarizing ryanidine. Ryanidine is the channels, so the channels close, then the ATPase wins. The tension also decreases now that cross bridges are not being formed because
Being
that's why cross bridges are not being formed.
What was the question?
Yeah, what
Why is tension dropping off? Cross bridges are not being formed, and why is the tension dropping off?
Motor units
ATP rebinds with the myosin head and then releases.
Well, yeah, that's when calcium is not present. Why is it going back to its resting length?
That was a bunch of quick dances machines. That's one.
Tighten and what else is helping the sarcomeres go back to the resting length? Tighten and series elastic components. So those include tendons. The connective tissue of the tendon is made up of endomysium and epimysium. Okay. Okay, so types of muscle contraction. Isometric. Every single muscle contraction begins with an isometric muscle contraction. It will remain isometric, meaning same external muscle length, until your force exceeds the load that is applied to the muscle. Isotonic is when the external length of the muscle can start to change. So it's apparent external length is changing. It says it's constant tension, but actually it's not really constant tension. At the full extension of biceps, when you're starting to generate force, there's actually more, it takes more force to get through this weak joint angle, less force to get through the mid-range, and then you'll pretty much run out of optimal force there. So it's not constant tension, but it is a moving change in muscle length is probably a better definition. Isokinetic, the velocity is constant. There is nothing in your body where velocity is constant. Isokinetic, you have to have some kind of external machine, and you've got to have that set up properly in order to get the velocity of the muscle contraction the same throughout the range of quick motion. You want to know about isokinetic machines, you go talk to Dr. Prince. He loves to talk about those, at least he used to. Okay. I don't know if they still, in the weightlifting, have isokinetic machines or not, but they're really bizarre to work out on. They feel really strange. So isometric, that external length that you see remains the same. But if you touch the muscle, you can tell that it's generating force. If you look at the sarcomeres are shortening, but that tension has not gotten to the point where it can then be shifted to the tendon, and you can't apply that muscle force to the bone, so the joints are not changing length. So external length of the muscle remains the same and will remain the same until the force that the muscle is generating can then start the joint to move. So my freaky friend here, creepy, every time I look at him, I'm freaked out by this guy's face. Anyway, let's forget his face and then look at the muscles here. The muscle here is biceps. It's attached to the radius, so that's why I know it's biceps. It only has one tendon, so that's kind of freaky as well. Should have two tendons there. The contractile elements, your actin and myosin, they will slide past each other and create tension in the muscle, and this force will, if the load is not moving, then you've generated just enough force to maintain the joint angle and not have the load extend the joint again. So the force is generating, that this muscle is generating here, is isometric muscle force. You're maintaining the external length of the muscle and the load is held constant. Grading contractions, you do that with varying the strength of the muscle as a whole, and you do that by recruiting individual motor units. So the number of muscle fibers contracting, you do that by recruiting more motor units or decreasing the number of motor units recruited if you want to lower an object down. You recruit more muscle fibers, then you can create more individual muscle tension added together, and if that tension exceeds the load, then the joint will change angle. Velocity of shortening, when there's no load applied, then you can shorten the muscles very quickly. So velocity of shortening is high when there's no external load applied to the muscle. When the external load is high, then the velocity of shortening may be 0. If there is no shortening, external muscle length shortening, then you're in an isometric contraction. Any point between this black dot and this black dot, you have an isotonic contraction. So you start the muscle to move and the load to move, then you are in an isotonic contraction until there's the velocity, the load is 0 and the contraction is entirely completed. So you look at your lever system and you look at the fulcrum, you look at the effort that's being applied, that's your muscle attachment, and then whatever the external load is, it might just be the body part, or it might be something like this 5-kilogram ball that was placed into the hand. Fulcrum is the joint, the pivot point of your joint. If you're looking at muscles like biceps, you have the insertion on the radius distal to the trochlea. So the effort is applied distal to the fulcrum. The load, the weight of the arm or the external load that's being applied, find the center of the load and then measure to the fulcrum. That's your your lever arm or load arm length. And then the effort arm is again from the fulcrum to wherever the biceps is inserting. So that's your power arm length. The lever ratio of 5 to 35 is a ratio of 1 to 7. This is designed for a lot of distance covered with muscle contraction, but not a lot of power that's created in this lever system. So this being what kind of lever? Fulcrum, effort, load. Third class lever. These are designed for moving over a large distance with a moderate amount of force applied, but it's more for velocity, the speed of the movement, as opposed to the absolute power that the muscle is applying to the joints. So motor units we've defined in Martini as one motor nerve. Specifically, what kind of nerve? No. Alpha motor neuron. Why? Because there also is a gamma. So we're going to be talking about alpha and gamma motor neurons. Alpha motor neurons run your motor units. One alpha motor neuron recruits X number of muscles, and that's your motor unit. If you want to produce precise, delicate movements like threading a needle, constricting, well, that's smooth muscle, so that's not a good idea.
Well,
But fine motor movements like writing or drawing, you have small motor units contracting so you can get precise movements. When you want power, then you recruit large motor units. You have one nerve recruit a lot of different muscle fibers, and maybe 15 to 2,000 muscle cells per alpha motor neuron, and you've got power muscles. The smaller motor units may contain just a couple, 2, 3 muscles, or maybe 12 or so, but less than 20 usually in a small, for precise fine motor movements. Gross motor movements, you're gonna heave.
oh no, the ball was.
Okay. We all have things that fascinate us. Okay, 1,500 to 2,000 fibers. Okay, so Martini had something similar to this with blue, red, and purple. Now we've got red, beigey, orangey things. So motor units are the alpha motor neuron that's up here, this nerve cell body, and the muscle fibers that it recruits. Asynchronous recruitment means that you're not firing all your motor units at the same time, but you recruit some, you recruit enough to do the work. As those are fatiguing, then you will select some other motor units in that muscle to turn on, so they take over the power generation as the other motor units are fatiguing. So you can maintain your power output by selectively recruiting different motor units. That's referred to as asynchronous recruitment. And you can keep the muscles acting for a longer period of time. When you are trying to maintain your posture throughout the day, you have selective recruitment of these erector spinae muscles so that your posture does not suddenly turn off and your head fall over or you fall over. So when you're doing fine motor movements, then you're selectively recruiting the smaller motor groups, and the overall muscle strength of muscle contraction remains low, but it has precision. When you're using your leg muscles, then the amount of power generated by each motor unit is larger, but you're not going to get the precision with these large motor units. So relative strength, large motor units, more power output, but less control, less precision. Smaller motor units, less power, but more precise movement. The whole muscle as a whole will be dependent on the frequency of the stimulus arriving. It will depend on the length of the muscle fiber at the onset of muscle contraction. If you overstretch a muscle by having some weight stretch on it, your zone of overlap will get smaller. So the number of cross bridges being formed will be less, and your power will drop off. If your muscle is already pre-shortened before the stimuli arrive, then you're running the risk of ramming myosin into the Z-disc and damaging your sarcomeres. So the body is set not to contract at those points, at that point in time when myosin might ram into the Z-disc and rupture the Z-disc. So power will drop off if your sarcomeres are already shortened too much. There will be no power generated or less power generated if there's fewer cross bridges that can be formed. So if you have optimum overlap of actin and myosin, you're going to get the optimum force production. Fatigue, there are various theories of fatigue. We'll cover some a little bit later. You're probably never going to run out of ATP. That's not the reason that you fatigue. You're probably not running out of acetylcholine, but there are other local events that are occurring that might interfere with cross-bridge formation. So it's fatigue. There are some good reasons for fatigue and some not so good theories of fatigue. When you hypertrophy the muscle fiber, which is the muscle cell, you put more cross bridges in parallel by making more myofibrils in the muscle cell, then you can generate more muscle tension. So the fatter your muscle cell is, assuming that it's getting adequate nutrition, then you can produce more power in that muscle cell. Like neurons, you can sum twitches together. If the stimulus arrives and the muscle cell goes through its contraction and there's no stimulus for a period of time, and then re-stimulate the muscle, if the muscle has come back to its resting length already, then there's no summation between the two twitches. But if you move the stimuli closer together so that the first contraction isn't finished before the next stimulus arrives, then you add tension together. So what do you think is happening here that is allowing the next twitch to be added to the first twitch? Why do you get an increase in force of contraction? Why don't you just get two identical peaks here?
More calcium.
More calcium where?
More calcium. Like you said.
Yeah. Too bad you can't work as a team, right? So calcium has not been pumped back into the SR before the next stimulus arrives. So some has, that's why the peak has come down a little bit, but then the next action potential, series of action potentials arrive, Ranitidine channels are reopened, and now you've still got about 40,000 to 1, maybe 39,000 to 1 of calcium to rush out again. More calcium in the cytoplasm, so more cross bridges form, tension goes up. If you run a whole series of action potentials through the muscles so that there's no time for any calcium to go back into the SR, you just keep those ranitidine channels open, then the muscle tension goes up nice and smoothly until all cross bridges possible have been formed. You maintain that power output, and that's referred to as tetanus. It is different than lockjaw, well, sort of, but it's similar, but it's not lockjaw that we're talking about here. We're talking about peak muscle tension. It will remain smooth as long as there's no time for calcium to go back in and come out of the SR. So the frequency of those action potentials have to be close enough that the ATPase never wins. There's never any depletion in calcium concentration in the cytoplasm. So once you turn off the action potential, or when internal fatigue or external fatigue occurs, then your tension drops off. So either action potentials are turned off and muscle relaxes, or action potentials are still firing, but other elements that lead to fatigue of the cross-bridge tension development take over and tension drops off. Okay, take a break. We'll talk about this length.